Ladies and gentlemen, welcome to this latest and last in the term teaching session in the course on technology and the future of medicine. Today, Nader is taking us through tissue engineering and regenerative medicine. Are we living in a science fiction? Take it away, Nader.
All right, thank you so much for being here. So my name is Nader. I am a master's student candidate in experimental surgery. And as Dr. Salah said, my topic will be about tissue engineering, regenerative medicine. Are we living in a science fiction? So to take it through my project, I will be starting off with the past, history about tissue engineering. So to start off with a quote, it is in the timbers of the past that the future is echoed. That really, for me, that quote represents that when we look into the past, we find that a lot of our present technological advancements have taken their precedent from myths. And throughout human evolution, it is evident that our capability to think has set us apart from animals and allowed us as humans to evolve to the top of the food chain. And the human brain has evolved over the course of history from thinking primarily as a hunter and gatherer to interstellar exploration nowadays. And it really shows that from the very beginning, like almost 2.5 million years ago, we had the first utilization of the creation of the stone tool. And that was pretty much the early transition, like human, which helped pretty much usher like creative thinking in our evolutionary ancestors. So since the dawn of time, humans have been thinking of ways to help them improve their standard of living. One can then argue that technological advancements have been the cornerstone of our maturation, progression as a race, and improving our standard of living by helping us achieve what was once considered mythical. So for instance, repairing, replacing, or regenerative cells, tissues, and organs to restore impaired function. It is these myths that are documented from the past which helped intelligent humans dare to think differently from the rest and create the impossible. So a famous philosopher in the early mid-1900s, Ayn Rand, stated the difference between animals and humans. Animals change themselves for the environment, but we change the environment for ourselves. So to jump a little bit ahead now into the Enlightenment Era, which is the late 17th to the end of the 18th century, we find that during this era, it's the scientific research in the fields have come very far since the Enlightenment Era. And whether it is the advance in anatomy, physiology, surgery, or the exciting field of tissue engineering and regenerative medicine, the Enlightenment Era has definitely emphasized empirical scientific research. And it was the push towards objectivity and a reasoned approach to study nature and the human body. And during this time or era, we found philosophers and scientists investigating the emergence of reproduction and regeneration to understand the origin and governing dynamics of life. So then the question is, what is tissue engineering and regenerative medicine? Well, it can be broken down into the two sets, the regenerative medicine part and the tissue engineering. Nowadays, it's synonymous. However, back then, regenerative medicine was seen as a translational research that applies findings from basic sciences into useful medical and nursing practices. In other words, this translational research is a broader field that includes tissue engineering and molecular biology, primarily focusing on the process of replacing, engineering, or regenerating human cells, tissues, or organs to establish and restore normal function. While tissue engineering, on the other hand, it primarily focuses on functional and preferably autologous, or in other words, from your own body, substitutes that can either improve or replace a failing tissue. Both ultimately try to alleviate certain healthcare problems which we face nowadays in our society, primarily shortage in organ transplantation donors and the rejection that occurs after organ transplantation. Nowadays, regenerative medicine and tissue engineering are both synonymously used together as almost the same thing. So in the past, tissue engineering has been applied to the use of prosthetic devices and surgical manipulation of tissues. It has evolved from the field of the biomaterial development and refers currently to the practice of combining scaffolds, cells, and biologically active molecules into functioning tissues. And regenerative medicine, that term has first appeared in 1992 in an article of Hospital Administration by Leland Kaiser. Kaiser's paper concludes with a bold statement saying regenerative medicine, pretty much like coining the term, where a new branch of medicine will develop that attempts to change the course of chronic disease and in many instances will regenerate tired and failing organ systems. So, as mentioned, that with that first quote, it is in these mythical documentations that we can look in the past to see how far tissue engineering and regenerative medicine has come. So, to briefly go into it, some of the terms or topics that are introduced in TERM, or tissue engineering and regenerative medicine, one of them is total potency. So total potency just refers to stem cells which are used in tissue engineering and regenerative medicine to divide and produce a specific differentiated cell of tissue. And the more cell types a cell can differentiate into, the greater its potency. So this concept was then first attributed to the Hindu myth of Rakta Baj, which Rakta means blood and Baj means seed, where Asura whose blood drops could form his clones ex vivo. And then another concept in tissue engineering and regenerative medicine is in vivo regeneration. We see this concept in documentations in the past being described in the Greek myth of Prometheus, who was a titan who stole from Zeus fire to offer it to humanity, and then he was sentenced to eternity of misery, where his liver would regenerate each day after being pecked away by an eagle to prolong his suffering. Therefore, once again, just showing us in myths the concepts of tissue engineering and regenerative medicine being prevalent. Another concept that we see in the past is bioreactor, which helps supports and stimulate development ex vivo. In an Indian epic Mahabharata, a story describing the birth of Kaurava brothers, the mother, after being pregnant for 2 years, produces a mass of flesh, which a sage takes and divides into 100 parts, then keeps it in a pot, where it treats it with herbs in the form of butter. And the combination of both then presumably results in a culture media for 2 additional years. And it is after the 2nd year that there is a rise of a viable human, or almost what we can think of nowadays as a clone, and 100 Kaurava brothers were born. So this is probably the oldest reference in human history, 400 BC, illustrating perfusion culture. Biomaterial is synthetic or natural material suitable for use in constructing artificial organs and prostheses to replace bone or tissue. So once again, we see this prevalent in around 3500 to 1800 BC, where Queen Veshpala was apparently amputated in battle, was then fitted with an iron leg to allow her to return to the battlefield. So now moving away from the mythical applications or the mythical documentations and entering, okay, now has tissue engineering been around in the past, and has it actually been like applied throughout the period of the Enlightenment era, and where has it gone? We see that the oldest documentation of the application of tissue engineering and regenerative medicine was in the Neolithic period around 10,000 BC, and it was dealing or being documenting pretty much the suturing materials that ranged tremendously. So it ranged from, you can have natural suturing, such as catgut by the Europeans, or biological, like South Africans and Indian subcontinents used biological or heads of large biting ants to be able to suture certain cuts. And then the last one by Egyptians, synthetic, linen, for instance. So classical examples of tissue engineering and regenerative medicine, we see that cell-supportive scaffold guiding regeneration was first attributed by the Egyptian, the Papyrus of Eber, around 1500 BC. It had a very peculiar way of treating skin wounds using lint, grease, and honey. Lint was then believed to be due to, because it has fiber scaffold that help promote wound regeneration. The honey was believed for antibiotic due to its hypertonicity, and then the grease was believed to be used as a barrier to the entry of pathogens. During the Enlightenment era, we see now a trend or a start of a progression of the tissue engineering and regenerative medicine concepts. So It is during this period that the milestone and the revolutionary of the birth and the progression of TERM that is prevalent. We see that one of the individuals that pioneered the thinking of tissue engineering and regenerative medicine was Sir Isaac Newton, where his thinking started going away from the mechanical phenomena that was preceding him into the idea where he says a quote that man is a complex psychochemical machine and any disease that a man encounters is due to a fault in that machine. From there, however, that we can still see that due to the infancy of tissue engineering and regenerative medicine and the concepts being there, philosophy's downfall couldn't really explain some of the complex phenomena that are nowadays can probably be explained a little bit better with more accuracy due to our technology and progression in knowledge about reproduction, regeneration, and even growth. Next, in the Enlightenment period progression, we have Benedict de Spinoza, and he was the man who is believed to have laid the foundation for modern tissue engineering and regenerative medicine. He wrote a letter describing the existence of homeostasis and dynamic crosstalk between various elements within the body. And then shortly after him, we see Ibrahim Tremblay conducting the most remarkable set of experiments during the 18th century, investigating regenerative capability in hydra. And on the side over here, these are the hydra, where he concluded in his article that anywhere he cut the hydra, regardless of the axes, it would remarkably regenerate into an entire organism from each piece. And this was very revolutionary because before he conducted this concept, the regeneration thinking was more that it was being considered by the whole organism compared to the cellular level, that the cells were causing regeneration. And then once again, as we go through the progression, we see that Henry Levert was continued, for instance, to conduct the efficacy and the safety of various metallic sutures in dogs. He laid, therefore, the foundation on what sutures are biomaterial, which ones will corrode within the body. And after a century later on, we have Arthur Zeller pretty much taking the information that Henry Levert founded, and he found that using iron and steel result in rapid corrosion, copper, aluminum, and zinc results in tissue discoloration, gold, silver, lead, and aluminum lack mechanical robustness. And then it was the advent of the stainless steel that was found to be the best biomaterial. Christian Pander hypothesized that the existence of tissue development on a dynamic interplay between cells and the surrounding microenvironment. He was therefore accredited with playing a major role in understanding the development of germ layers and responsible for the generation of the organ systems of organisms. And from there, he is known, he has kind of set the foundation of the idea of how regenerative medicine can occur and how cells interact with each other in order to produce an organism overall. Then 40 years later, there was a proposal that tissue regeneration depended on cellular proliferation. And from that, Leo Loeb in 1897, he pioneered the growing of cells ex vivo. And then, however, it wasn't until 60 years later that the proving of stem cells or precursor cells surfaced in literature. So when we really look at some of the experiments as we discuss about the hydra and the suturing examples and everything, we find that one of the first ex vivo bioreactor or perfusion circuit was conducted by Alex Carroll and Charles Lindbergh, where they created a continuous perfusion flask, which provides a tightly controlled environment to sustain organs ex vivo. Yet it was still capable of perfusing them and allowing the organ to stay alive and remain in good conditions for at least 3 weeks and in usable conditions for at least 1 or 2 months. And that is very remarkable, especially during that time. And this was the flask that they created that resulted in a sterile condition. So now, based on that's the history, and all of these individuals have influenced us to get us to where tissue engineering and regenerative medicine is nowadays. Now we will start explaining about what are the modern applications that we see nowadays with tissue engineering. So to begin, stem cells is a huge thing when it comes down to tissue engineering and regenerative medicine. They're unspecialized cells. They can either replicate themselves or form a variety of special cells via a process known as differentiation, as shown in this diagram. And then the differentiated cells can then go on to produce a variety of special cells. And then those cells can serve as a repair system in case dysfunction occurs within an organ. So where do stem cells come from? Well, it's primarily two places right now. It's either embryonic stem cells or adult stem cells, also known as somatic cells. So the adult cells can also have induced pluripotent stem cells, which have recently been discovered around like 2007 and 2006 for mouse and human respectively. And these induced pluripotent stem cells are genetically altered reprogrammed cells that act in a way like embryonic stem cells. However, unlike embryonic stem cells, they don't result in a lot of the cons that embryonic cells do after transplantation, such as formation of tumor cells, as well as some rejections that can occur. So the difference now between when we compare embryonic stem cells versus adult stem cells, we look at adult stem cells, and originally they were only believed to come from bone marrow. However, scientists have now discovered that stem cells actually exist in many different organs. And some of the pros with using adult stem cells is that they remain in their respective organs and don't divide or differentiate until necessary for repair, worn out, or diseased tissue. While embryonic stem cells are known as pluripotent cells, and under the right circumstances and factors, embryonic stem cells in culture will also regenerate without specializing in tissue. And then within the body, they cluster together and then they form an embryo body, which eventually differentiates into tissues in an organ and an organism. So some of the pros of using embryonic stem cells is that they are pluripotent cells, as we discussed, and they can differentiate into a variety of tissue and organs. And then, however, some of the cons with using embryonic stem cells is yet that there hasn't been any really safe medical use found for embryonic stem cell usage. And there's always going to be an ethical concern because scientists and other individuals will believe that like any of those pluripotent cells could grow into becoming an organism or a human being. Therefore, if you are taking a cell out, are you technically killing a human? And then as well as the more important thing is that they pose a serious threat of forming tumors because of their pluripotency. And in transplantation, patient organs may reject the stem cells and they can result in being permanently trapped within the body. Now, unlike adult stem cells, which the majority of regenerative medicine is currently using in tissue engineering, they're mature and genetically stable. Therefore, we don't have to worry about tumors being formed after transplantation. And they are currently the only type of stem cell commonly used to treat human diseases. And adult or induced pluripotent stem cells are taken from the patient's own body and thus no risk of immunorejection post-transplantation. And then a couple more things. There's actually 73 human diseases that adult stem cells can help alleviate. However, some of the cons is that when we try to use them in bulk for tissue engineering ex vivo, they're restricted in division once they are removed from the body. And this is a brief diagram of them being removed from the bone, being cultured, and the important cells that we want to differentiate. And same thing with embryonic stem cells, just being called pluripotent. So is there an alternative to stem cells then? Well, currently there's amniotic stem cells and the umbilical cord blood stem cells. Both of them, as their name describes, that's where they come from. And both can be utilized, and they're very similar in embryonic stem cells. However, they don't carry the ethical issues that embryonic stem cells do. So when we look especially at umbilical cord stem cells, there is a lot of research going into them and figuring out, like, are they useful? And based on the literature that is present in everything, there is approximately 4 million samples of cord blood that are discarded in hospital nurseries every year. That cord blood actually contains a lot of stem cells, which then can be used actually for like transplanting as much as 80 serious medical problems and alleviating those problems. Those can include prevalent types of leukemia, metabolic disorders, or blood-related conditions such as sickle cell anemia or severe anemia problems. And then some other treatments that these umbilical cord stem cells can actually help alleviate in today's society: heart attack, Parkinson's, Alzheimer's disease. So some of the examples nowadays that when we look for stem cell therapy, we see that some of the literature that has gone out has been using mice and other animals, taking their bone marrow, the stem cells from their bone marrow, and actually transplanting them in damaged heart, right here. And then they find two things can happen. Either, like this was actually also conducted, like a few studies carried this out in human patients, with patients undergoing open heart surgery. They found that the stem cells injected in the circulation into the injured heart resulted in an improvement in the cardiac function or induced the formation of capillaries. And this is what the diagram is representing. So another, why did it delete? So one of the greatest actually breakthroughs in some of the present therapy for stem cell was conducted in 1997 by Dr. Charles Vacanti. There is actually supposed to be a picture here. For some reason it's on my computer, but it's not showing up. So the breakthrough was that he was able to actually obtain like a mesh of wire and a scaffold and pretty much grow an ear on the back of a genetically engineered mouse. And this mouse was known as the Archaeolosaurus mouse, or the human ear-bearing mouse. And this was really the first time that a synthetic tissue can be created from cow and sheep cells on a scaffold and produce a full-sized ear on a rat model. And like the intention for this, like why it's so groundbreaking, is that like individuals with like burn victims, for instance, and for plastic surgery, now we have another way of obtaining those ears or noses that they've left using their stem cells, therefore resulting in no rejection at all after they're being implanted. So further on, now we fast forward from 1997, using the same concept from Dr. Vacanti. In 2012, Dr. Patrick Barnes, actually, from Johns Hopkins, was capable of attaching to a patient one of her ears that she lost due to cancer from her own tissue. And as you can see from this diagram, this is what it looked like. Like she had, I believe, a basal cell cancer, which resulted in her ear canal and their ear to disintegrate pretty much, or become dysfunctional. And therefore, he took the cartilage from the new ear, which was derived from the patient's ribs, matched it, and then he was able to implement it in the arm for growth. So taking that, putting it into the arm, those two things were for perfusing the ear to grow. And then after, surprisingly, she actually grew an ear, and then he re-implanted it with 0 rejection. So now when we move on to organ, tissue, and engineering and regenerative medicine, we were talking about the stem cell or the cell line. Now we're going to move up a little bit and start talking about the organ and where tissue engineering and regenerative medicine is taking us with. So there's a huge shortage of donors currently right now. As of 2012, there's approximately 2,000 transplants that have been performed Canada-wide. However, the number of individuals on a waiting list waiting for just a transplant, whether it's kidney, heart, lung, liver, pancreas, is around 4,500. So therefore, that number is going to be continuing to increase a lot faster than the transplantation that are available. And from that, we see that 250 individuals died in 2012 just waiting for an organ. And from that, then a third of those individuals who died were waiting for a kidney transplant, ushering us or showing us how important, for instance, that we need to find an alternative to the shortage of donors, especially for kidneys. So the alternative solution then to transplantation is tissue engineering and regenerative medicine. It will usher a new era of therapy and alleviate the high mortality rate currently present due to the lack of organs. The technology will continue to progress improving, for instance, like 3D printers, bioreactors, and further understanding how to use differentiate stem cells in order to create whole tissues and organs ex vivo or outside of the body. While TERM, or tissue engineering regenerative medicine, it is still decades away from producing like a viable clone or a full body as it was described in that myth. However, creating a full functioning, however the technology, exists to develop available functional tissues and recently organs outside of the body for clinical applications in the future. So now the question comes down to science fiction or real? Like, is the stuff that we're reading on the news, is it real or not? And it's hard to believe when you think back to where we were in the Enlightenment era, where they were hardly understanding how stem cells were able to regenerate or what they even were, to nowadays that we have heart being bioengineered in labs. Yes, they're far away from clinical applications, but it's impressive that they're able to take the experiment, they're able to decellularize, for instance, a donor rat heart, get the scaffold, and then from there use it as a scaffold, add to it autologous cardiac or endothelial cells, and then reconstruct a heart, which eventually showed contractions. So is it okay if I can just quickly play the YouTube? Now, on a similar premise, we saw the heart. They're currently actually being able to use the 3D printers that we have to print a human kidney, due to the idea of that kidney transplants or the waiting list for kidneys are a lot larger, and there's a lot of deaths for a kidney waiting list compared to heart, lung, and all the other organs. So I am not going to be showing the video for that, but it's the same idea for the kidneys. They're taking, for instance, a complex organ, and even though it is a complex organ, there hasn't been any development for clinical use yet, but they take the same idea. They take the kidney, they decellularize it with a detergent. From there, they're actually able to obtain a scaffold, whether it's from porcine, a human kidney, or a rat, take the scaffold, then recellularize it. And what they found is that it yielded a bioartificial kidney with very basic urinary generation function in vitro, which is still remarkable to see how far we've come. And then when they orthotopically transplant it back, so putting it back into the rat where anatomically it belongs, the graft then becomes perfused and the recipient circulation is able to produce urine in vivo, which indicates a functioning kidney after producing it from a 3D printer. Now, on the same scale, lung bioengineering, it's the exact same thing, they do the exact same thing, and remarkably, they're actually, the kidney lab was able to incubate these cells in a bioreactor and yield and construct a lung that is able to exchange gas in vitro at a level comparable to native lungs. So they're almost as good as native lungs. And to demonstrate the lung function in vivo, once again, they're transplanted back in vivo, and they found, once again, that the gas exchange was able to go up to 6 hours after removing the tracheal tube. And then similar to the kidneys, though, that these tissue-engineered lungs or the kidneys yet have been transplanted into humans. But that is where we see the progression going towards. So moving away from now the bioengineering aspect, regenerative medicine isn't just about creating organs as well. Like, there's other solutions right now, something that I'm currently working on. For instance, the ex vivo lung perfusion system, as we saw in the past, in the Enlightenment period, where Lindbergh was able to create that perfusion circuit. So that on its own is still a sort of regenerative medicine. Why? Because we have now this technology which is in its infancy, just been around since like probably 2000, like actually being pioneered by 16, at least for the lung, where you have a circuit where a dome where we can place the lungs in there, have it perfusing with normal physiological conditions, and then be able to repair and hopefully regenerate injured lungs over a perfusion time of 12 hours if we understand what kind of perfusate solutions we need in order to have those lungs outside of the body for 12 hours. So this is what the schematic of this circuit, of the perfusate circuit, looks like, and then this is what our lab is currently working on. And it is being seen that the gold standard in the past of just taking organs and placing them on ice to hopefully slow down the cell death and as well as the metabolic demands of cells, that is going to be changing over the years, and implementing a portable ex vivo lung perfusion system, for instance, or a circuit, in order to allow assessment of function or deplete that ischemia period or lack of blood that these organs occur once they're procured out. We place them on these circuits and we maintain them in normal physiological conditions throughout the transport period to the recipient site. And therefore, during that transportation, we're able to assess the lungs for their functions. So therefore, we're not transplanting bad or injured lungs as in the past we used to when they were on ice. And on top of that, we can actually repair or regenerate the lungs if we find them unsuitable for transplantation. So, future of medicine and the final remarks then. Well, we know that the technological singularity is approaching as the years pass. Why? Because of how we've seen since the 18th or 17th century, where we hardly really knew anything about stem cells, and nowadays we're able to create lungs, hearts, and kidneys through printers or in the lab. So our technology and our understanding based on technological advancements is increasing at an exponential rate. So we are approaching that. So as we approach that then, it is going to be expanding exponentially. And as the years pass, there's going to be more groundbreaking technologies that the technology, for instance, that was 5 or 10 years ago will become obsolete in a few years. And this is evident nowadays by seeing, for instance, video game consoles. If you want to look at that, like how much that has changed over the years in such a short span of time. Communication devices, looking at your iPhone compared to like a Motorola or a Nokia phone back in the day, like 5 or probably like 10 years ago, medical devices. And remarkably, even though 3D printers have been out only for like 30 years, we're starting to see a recent shift that's going to happen in 3D printing as well. So 3D printing, as mentioned, has been around since the 1980s. But however, recently in March 2015, Joseph de Simone, the CEO of Carbon 3D Printing, introduced a way of printing material, like a new way of printing material 3D. And they were inspired by, believe it or not, the Hollywood movie of Terminator 2, where the scene of T-1000 just emerges from a pool of liquid and forms like a functioning body, I guess. So they wanted to produce a 3D printer that operates the same way. So using a new technique referred to as the continuous liquid interface production, or CLIP, the technique combines pulse of light, which solidifies the resin, and oxygen, which prevents it from solidifying, to craft an intricate 3-dimensional object. This works continuously, unlike current printers that construct objects by building layer upon layer. And unlike current 3D printers as well, that construct layer by layer, this is a very, we can use better quality for that. It's more durable because you don't have certain tensile forces, where if you're building on a 2D dimension, layer by layer, at certain angles, it'll actually be weaker compared to a printer that continuously creates an object as it's pulling it out of a pool. And the biggest impressive thing about these carbon 3D printers is that they can actually create an object which these current 3D printers would take around 6 or 10 hours in approximately 7 minutes, 25 to 100 times faster. So it's just a brief 2-minute video just to show how. All right, so therefore the future now. So based on all this technology, like what do we see tissue engineering and regenerative medicine going? Well, we can try to extrapolate a little bit based on the progression that we've seen. And what I personally believe, and I believe will happen, is that in probably 50 or 100 years, I believe that we might start seeing organ donation banks or facilities. And that, in my opinion, is a plausible scenario based on how far we've come so far and how fast our technology is changing. Take, for instance, like I said, the carbon 3D printer that now can print things 25 to 100 times faster. What if that technology continues to progress, which it most likely will continue to progress because we're on an exponential curve here. So when we look at the 2005 sci-fi thriller, The Island by Michael Bay, the premise really of that movie was that you had people who wanted to live forever, and how they did that was by growing clones, and they had the clones there in case, for instance, they needed organs, so they would just harvest their organs. But unfortunately, I personally think that tissue engineering and regenerative medicine is still ages away from being able to produce a full viable clone, a human. However, we're not too far-fetched of having organ donation facilities because we're currently already producing organs from printers as well as in the lab. So the technology, once again, like I said, of carbon 3D being implemented and improved in for bioprinters will really usher a new era. I believe in like the whole bioreactors and how organs are being produced outside of the body, body. And then organs in the future can be given to you in a matter of minutes, if not hours now, with this improvement in carbon 3D if that's implemented in bioprinting. And you won't see any transplant waiting list or mortality rate anymore because all you have to do really is to go to one of these facilities, give them either a DNA or just a couple stem cells, and with the technology that we'll have, they'll give you the organ that you need. So therefore, we should see no more post-transplantation implications due to zero immunoreactivity because you're receiving your organ 100% DNA. And then the ultimate goal then, in my opinion, is for us to somehow be able to regenerate our consciousness, or at least be able to remove our consciousness from this body that we're in and be able to take it and place it into another form or another functioning body. And this actually was inspired, or it inspired me to think about that because this movie that recently came out called Chappie, it's pretty much a robot that has been designed or incorporated with a program where somehow he develops full intelligence, or full artificial intelligence, where he's able to be conscious and therefore alive. And from there, he was able to answer the questions and solve for us, as we mentioned with technological singularity, that once we have full AI, for instance, they will be able to solve that ultimate goal where what is consciousness? Can we quantify it, and can we take it out and put it somewhere else? And therefore live forever, or live in a body that doesn't need maintenance or doesn't break down because our organs keep aging. So as mentioned then by Albert Camus, man is the only creature who refuses to be what he is. And that's a very interesting quote in my opinion because as to what we are supposed to become or will become in the upcoming years, no one can really foresee such a thing. However, approaching certain ailments and shortcomings in the future, such as organ donation like shortage or chronic debilitating diseases, will be viewed and treated differently, in my opinion, in like the next 50 years. So some might no longer exist or pose such a threat in our medical communities, and that's due to the advancement in the field of tissue engineering and regenerative medicine that will continue to progress and continue to improve. So what I think is that what we consider normal therapy nowadays would have definitely been viewed as science fiction 50 or 100 years in the past. So therefore, are we living in a science fiction? I believe that we are living in the science fiction era that in the past they would have viewed that. However, nowadays we see that as the norm, and who really knows what in the future what will become like the normal back like in 100 years. So that would have never been possible though if those enlightened individuals refused to think differently than what was deemed norm or acceptable during their time. And to finish off with a quote, without deviation from the norm, progress is not possible. It can have definitely a positive or a negative ramification on what is still to come or to be discovered. Thank you.
So I find this very interesting, Nader. I wanted to point out that in the paper, you kind of lead up to this final table, and the reference for that table is very weak. So, and the same thing kind of happens in the presentation. When you talk about 53 diseases that can be treated by stem cell. So in table 1 in the paper, the reference for that, reference 56, which is donoharm.com, that isn't a real site. It's just a series of links. It's kind of a placeholder site.
Okay.
So what I'd like you to do is to go into that site and figure out which link from it you actually used.
Oh, maybe.
Because the site itself is not a sort of credible site.
No, yeah, of course. I probably just like
right.
wrote
So figure out which link, and then how credible is this list?
Yeah.
And is the fact that there are 53 entities on the adult side and none on the embryonic side, is that a kind of even-handed decision, or was this a site put together by people who are, heavily biased?
Yeah.
So that's the one thing I would like you to add is to tune up reference 56 to a real site with real authors, real sources, and then figure out how rigorous this list of things that can be cured by adult stem cells is.
Well, like on top of that reference number 56, like I didn't include it in there, but like the national, like one of my references was from the National Institute of Health, and that was, it had pretty much something very similar to what they were, like obviously not the exact same number like you're mentioning, but they were also mentioning how embryonic stem cells are hardly used nowadays for transplantation because of the cons that they show compared to adult stem cells.
Yeah, no, I'm certain that you'll be able to find
concrete,
rigorous, concrete lists, and the paper would be stronger, I think, if it sort of ended with that rather than with this nebulous thing. Yeah. It's really exciting, the definitive start of both regenerative medicine in '92 and tissue engineering, which is usually considered to have started in 1988. Yeah. Now, if you look at the scaffolds, there's a literature on the tissue reaction to them. It goes back to '69. Yeah. And so all of those are rather definitive beginnings. Before '69, there wasn't anything. Before '88, there wasn't anything. And before '92, there wasn't regenerative medicine. So this is a very new field.
It is, yeah.
And pathologists are authors on both of those papers from '69 and '88. So in that sense, the kind of morphology of the situation was, important from the very beginning. Yeah. Do you have any questions?
Not a question, but perhaps a comment. Had you been doing this paper maybe 5 or 6 or 10 years ago, I think you might have been doing an awful lot more on cloning and on Dolly. And I guess I was somewhat surprised that you made no mention of, I guess, the failure of the Dolly experiment.
Yeah.
And what implication that might have for tissue regeneration and in fact the whole regenerative field. So I just wonder what your response to that would be.
Yeah, it completely, now that you mentioned it, actually completely just slipped my mind that I remember even reading it back in the day. And it was, back in the day at least, it was groundbreaking stuff, because during that time, it almost actually relates to a little bit to the whole embryonic stem cells, and how, especially when the mouse came out and individuals saw like, whoa, like there's an ear on a mouse right now. And then you start going into the whole concept of genetic engineering and how people didn't really like the whole idea of genetic engineering. You're playing God. And same thing with the dolly. Like, I guess, like, it just slipped my mind, honestly, for not including it. But the fact that it failed, I think honestly, it just helped us focus a little bit better on the idea of like not focusing too much on cloning or like cloning organisms or humans, and focusing more on the idea of, okay, let's start a little bit smaller than that.
Yeah, and there's another issue that I see, and that is that this area tends to be hype. You know, there seems to be a media dimension to this that really is not part of the original kind of scientific idea. And so there are now papers that are written which is highly critical of the kind of razzmatazz that's behind this thing. And so I'm very interested in what I would call the restraint of science in developing this area, where it looks on the one hand very promising, but on the other hand may end up another dead end, you know.
So what one can say in terms of tangible progress, there was a group that predicted that they would have a functioning mouse thyroid generated from stem cells in 2015, and they succeeded.
Yeah.
You know, they did it. It's there. The same group is predicting that they'll have a functioning kidney from stem cells in a mouse in 2018. And you know, if you're a politician thinking in 3 or 4 years cycles, to predict that something's going to occur in 3 years is really much more meaningful than talking about 10 or 20 or 50. Yeah. You know, it becomes something that is tangible and you can plan around. So I think there are non-hype areas here where real progress is being made. And if you want to look at links to this course, like Shuvo Roy, he said when we Skyped with him on March 1st that the clinical trials of the bioengineered kidney that have a sort of regenerative medicine part, because there's a bioreactor part, would be starting in a year and a half. So that would be late next year, 2016. So there are real things happening, and I think
to add a little bit to that, like I agree with you that this field, especially, like it's very promising, but at the same time, like based on the media and how much the media influences a lot today, that the media can kind of like fabricate a little bit the information or mislead the average Joe, I guess, who doesn't really read the articles or anything of, like you said, for instance, like a website like that with a bunch of stated like diseases that would cure, but obviously like if you look into it a little bit more, like I know throughout the paper I didn't include all of those, I actually included from other like valid references, is that I was amazed myself, and I guess like I got misleaded by like having that. But then as Dr. Salaz is saying, that there are some parts where they are being like a little bit over the top with it, and maybe a little bit kind of ridiculous, because in the span of like what they're saying, like even the kidneys, like yes, like it is something tangible and something that you can work around in 3 years to have something like a kidney that's functioning and like you can maybe transplant it back. But in my opinion, I think that's still far a little bit, because I don't think our technology is still ready for it. Like yes, they have the bioreactors and they were able to produce it, but like what I've shown here, the kidneys are still, like they're hardly producing urine. So
just one other thought, and that is that it may be easier to build an ear than a brain. For example, the disease of Alzheimer's or Parkinson's seem to me to be far more complex an issue to deal with than For example, dealing with an organ.
An ear that just has cartilage, for instance, yeah.
So I think the non-hype areas here can be hard to discern, but what would be good, I think, about fixing table 1 is, you know, to end with something credible, tangible, rigorous. I think that's really what you need. That you don't want to contribute to the hype. You want to be part of the reality.
I personally even mean it as to end with it. I was meaning it as it's just a supplemental table. But when you actually read the essay, I actually include only specific things that like journal articles have said like they actually like deal with and work with. But yeah, I should have really not put that in there.
No, I think it's fixable. So, you know, come up with a reference for this, or come up with a more rigorously obtained, you know, table. I think that
that's what I'll do.
Good. Okay. Well, I think that's probably it because time is going on. So, thank you very much.